Dear Sjöberg laureates, dear members of the Sjöberg family, esteemed colleagues and friends, it is a great honor to introduce our second laureate, Professor James Allison, or Jim Allison, as he's known in the immunological community. According to himself, Jim was a curious kid, a good base if you want to go into science. In his case, this was facilitated by a number of circumstances: a supporting father, a chemistry set in the garage, good teachers, and a summer program at University of Texas after his junior high school year. And I will try to briefly review his career trajectory since then. He earned his bachelor's degree in '69 and his doctorate in biological science in '73, both from the University of Austin, Texas, Austin. After a postdoc at Scripps Institute in La Jolla, his first faculty appointment was at the Smithville campus of the University of Texas MD Anderson Cancer Center. In 1985, he joined the University of California, Berkeley, as a professor in the Department of Molecular and Cellular Biology, and also as a director of the Cancer Research Laboratory. He also served there as a head of Division of Immunology from '89 to '97. He was then recruited to Memorial Sloan Kettering Cancer Center in New York as the chair of the immunology program and as director of the Ludwig Center for Cancer Immunotherapy. In 2012, he moved to the University of Texas MD Anderson Cancer Center, this time in Houston, where he currently serves as professor and chairman of the Department of Immunology and as executive director of the immunotherapy platform. Now, Jim Allison presents an outstanding lifetime achievement in immunological research. He is particularly renowned for his contributions to the knowledge of T-cell function. As he puts it himself, he early on became totally hooked on understanding T-cell activation. In the early '80s, he pioneered the use of monoclonal antibodies as a discovery tool to try to identify the elusive T-cell antigen receptor. He produced one of the first, perhaps the first, antibody to a clonotypic cell surface molecule that later was confirmed to be the T-cell receptor. He then contributed considerably, and among other things, he contributed to the understanding of alpha-beta as well as gamma-delta T-cell receptors. In the early '90s, Allison investigated the molecular mechanisms behind the second signal required for T-cell activation, the one after the T-cell antigen receptor, in what was then an emergent field of research, the field of co-stimulation. A number of skilled immunologists worked in this field and contributed, of course. But Jim Allison was, together with Peter Lindsley, one of the first to explore co-stimulation in the context of tumor immunology. In 1995, Jim Allison's and Jeff Bluestone's groups independently showed that CTLA-4, a cell surface molecule on T cells, acts as an inhibitory molecule to restrict the responses of T cells. Building on this basic science discovery, and I would like to echo Bengt Westermark's introduction to Tony Hunter, never underestimate basic science discoveries. Now, building on this basic discovery, Allison, together with Dana Leach and Matthew Crummell, went on to do the critical tumor experiment, and that was to block this inhibitory molecule in tumor-bearing mice. And on a personal note, I must stop here and reflect, because this experiment, to me as an immunologist, if I or somebody in my group had come up with it in a group meeting, I would have thought of a dozen reasons why one should not do it. A number of reasons why it would never work, this strategy. But Jim Allison apparently thought otherwise. They went on to do the experiment, and not only that, over the Christmas '94, there was a blinded repeat of the experiment, and during this Christmas vacation, Jim Allison read out the results himself. And they were dramatic, and in a paper in Science 1996, they reported that antibody blockade of CTLA-4 in mice could lead to enhanced anti-tumor immune responses and tumor rejection. But he didn't stop there. All of this made him eager to produce an antibody to test also in humans. He tried to convince various large companies, and finally a small biotech company was bold enough to try. It would take over a decade from then to refine his discovery into a revolutionary pharmaceutical. And Jim Allison was an active participant on this journey. And to make a long story short, the concept of blocking T-cell inhibitory pathways as a way of unleashing anti-tumor immune responses was not only translated into one clinically useful antibody, ipilimumab, it also laid the foundation for the development of other drugs that target T-cell inhibition, which have been labeled as immune checkpoint therapies. Ipilimumab was approved in 2011 for the treatment of metastatic melanoma, and it is now explored also by itself and in combination with other checkpoint blockers in several other tumors. And the effect of these treatments have been dramatic for subgroups of patients with advanced disease, impossible to treat with any of the conventional cancer therapies. Jim Allison is a member of the US National Academy of Sciences and several other distinguished scientific associations. He is the director of the Scientific Advisor Council of the Cancer Research Institute in New York. He has received more than a dozen prestigious awards, for example, the William Coley Award, the Lifetime Achievement Award from the American Association of Immunologists, the Novartis Prize for Clinical Immunology, the Canada Gardner International Award, the Breakthrough Prize in Life Sciences, and the Lasker DeBakey Clinical Medical Research Award. And finally, what you may not know about Jim Allison is that he has played the harmonica for a blues band of immunologists and oncologists called the Checkpoints. And he also plays with a local band called the Checkmates. We now look forward to hear his lecture, Immune Checkpoint Blockade in Cancer Therapy, Prospects for Cures.
Thank you so much for that kind introduction.
Mr. Lower.
Okay. Thank you. I'm going to thank first the committee for letting me share this Sjöberg Award with my friend and colleague Tony Hunter. I think that covering the two sides of cancer therapy is really a great honor to participate in this award for this kind of progress on two different fronts of cancer therapy. I do wish that Mr. Schoenberger had made it to this meeting. It's a shame that he passed away just before this meeting. And what I want to do today is talk to you about the development of this type of drug called checkpoint blockade. I'm going to first start with where we are now and then go back through a bit of history so that you'll understand where it came from, and then talk to you a little bit about what's going on now. It's an incredibly exciting field. And I'd like to say first that like Tony, I was trained in biochemistry, and also like Tony, I didn't get into this work to try to cure cancer. I got into it because, as you just heard, I was fascinated by T cells. They were just discovered when I was in graduate school, and though my professor said he didn't believe that there was such a thing, he was an antibody guy. He really got me thinking about these cells that could go all over your body and look for things that shouldn't be there and kill them and protect you from diseases and possibly cancer, and not kill you in the process was something that you know, we're still struggling with. But I dropped everything I was doing at that time and started working on T cells. But then, of course, I was very aware of cancer. I lost my mother, 2 uncles, and my brother to cancer. And so whenever I learn something in immunology, I sit back for a while and think about it and try to figure out if it can be used to make a difference. So it's the immunology comes first and the cancer work second. So these are some relevant disclosures that I need to show you. And I want to thank, I'll thank the people who helped in this work. Jane Gross and Fiona Harning were the ones as graduate students in my lab identified CD28 as the major co-stimulatory molecule. As you heard, Max Krummel was the one in my lab who showed that CTLA-4 was an inhibitory molecule. Dana helped with the first tumor experiments, and then the other people later on with development of some of the stuff. I'm going to show today some work from Spencer Way showing how we can identify the cells that are responsible for tumor rejection. And of course, Padme Sharma, who's my partner, and with her we co-direct the platform for immunotherapy at MD Anderson. And then Metarex, who, as you heard, was a small company that gave helped us get this into the clinic after about 3 years of searching and being told that this would never work, that immunotherapy was rubbish, basically what I heard most of the time. Any bit, why immunotherapy? Well, theoretically, it makes a lot of sense because, first of all, there's specificity. And I think everybody knows that it's pretty specific. T cells recognize peptides in the groove of the MHC molecules. But what I mean here is that T cells have specificity. It turns out, this is something that Bert Vogelstein and I predicted about 2006, looking at some early genomic sequence data he had. The T cells actually recognize, in large part, peptides generated from the mutations that occur as a result of the carcinogenesis process itself when they're displayed on MHC molecules. And so I don't mean that they're specific for any particular antigen because it really doesn't make any difference, but they recognize the process of cancer formation itself by the formation of these active T cells. The second one is memory. Once you've got T cells, you've got them for the rest of your life. And so if the tumor comes back, you can reactivate them even more easily and eliminate it. And so adaptability is quite important too, given that genomic instability is what lets tumor cells escape many of the genomically targeted things. You can only see one molecule. The T cells, you've got somewhere around 50 million or so circulating in your body that can recognize that many antigens. I think that is a match for the genomic instability that one observes in differentiation of tumor cells. And so where are we now? Well, in 2011, the anti-CTLA-4 antibody that I helped develop was approved by the FDA for the treatment of metastatic melanoma. It was 3 years before the next approvals, again for 2 different PD-1 antibodies, again for the treatment of melanoma. In 2015, there were 5 approvals, and I'm not going to go through all of them, but again for lung cancer, adjuvant melanoma, renal cell carcinoma. In 16, there were 5 approvals also for Hodgkin's lymphoma, bladder, head and neck cancer, and non-small cell lung cancer. And this year, there have already been 3 approvals, again, non-small cell lung cancer, combination of, sorry, where's 2017? Well, this is not the most updated slide I had there. Anyway, there were 2 more approvals, one for Merkel cell cancer and the other was another one for head and neck. So 3 this year already. So there have been 15 approvals for these drugs in treatment alone or in combination in treatment of various kinds of cancer. And it's just a geometric expansion. I think we're probably going to have a lot more than 5 this year. There's 700 trials right now underway in PD-1 antibodies alone. So what is checkpoint blockade? Well, it's pretty unique because it doesn't target tumor cells and it doesn't involve vaccines or anything to turn on an immune response. And it works, as you heard, by blocking inhibitory pathways to unleash T cells to do what they were going to do anyway. And it came, again, as mentioned earlier, from basic science, basically trying to understand fundamental aspects of T cell activation. And so this cartoon kind of illustrates I can find this, what goes on. In the early 80s, we thought that engagement of the T cell receptor would be enough to activate T cells. By the late 80s, it became clear that that was not enough, that there were these mysterious co-stimulatory signals that could only be provided by dendritic cells and not by solid tumor, by other kinds of cells, but a very restricted kind of hematopoietic cell called a dendritic cell that expresses these ligands that were unknown. But we showed that the receptor was this molecule CD28. And the ligands were these, Peter Lindsley showed the ligands were these B7 molecules, they're called. CTLA-4 is highly homologous to CD28, and there was a great rush to figure out what CTLA-4 did. And it was first proposed to be another co-stimulatory molecule, but as you heard, Jeff Bluestone's lab and mine at about the same time independently found that it was actually an inhibitory molecule that stops T cell proliferation. And if you think about the biology of T cells, you have to have something like that because resting T cells with all this different specificity, this huge repertoire floating around, and when a virus comes in or something that's foreign that needs to be dealt with, you activate just a few cells. And so they've got to reproduce and give you the army out in the field to do anything. And what happens is you go from a dozen cells maybe to several 100,000 cells in the period of a week or so. So at that point, the T cells are dividing about every 6 hours or so. And you've got to stop that or it'll kill you. And at first it was thought that just activation itself would kill the cells, activation induce cell death. But now we know that there's actually this built-in inhibitory pathway that does it. So anyway, a T cell is activated when you get a contemporaneous recognition of peptide MHC complexes by the antigen receptor, and then recognition of the B7 molecules by CD28. That turns on all this stuff, the program that tells a T cell to start dividing, acquire effector function, to go, kill stuff, eliminate the problem. But as we know now, it also, at that time, initiates the seeds of its stopping, because it turns on the CTLA-4 gene. CTLA-4 then can accumulate with time until it reaches levels where it can outcompete CD28. It has an avidity of about 3 logs more than CD28 for the same molecules, and so it can just stop any co-stimulation by outcompeting them. But this is just turned on when you activate a cell. It follows that inevitably. So what does this have to do with cancer? Well, we'd done some earlier experiments before this with tumors. And we did several mouse transplantable tumor lines that would grow and kill mice. And I wondered what would happen. Obviously, they didn't have any co-stimulatory ligands. They would not co-stimulate. They would not present antigens to T cells. But we just transduced them with the B7 molecules. Those tumors got immediately rejected. And so the point is that they had plenty of antigens in them, but they were basically invisible to the immune system. And so putting all this in context, the idea I had was that, again, the tumor's invisible to the immune system, it takes off. No immune recognition until there's some tumor cell death at sufficient levels to call inflammation, and have these phagocytic dendritic cells come in and pick up bits of the tumor antigens, reprocess them on their surface, and then initiate the immune response. But then that also turns on CTLA-4. And then there's a race. If the T cells can eliminate the tumors before the CTLA-4 comes on, the immune system wins, and you might not even know you had a tumor. On the other hand, if the tumor wins, if that CTLA-4 turns off the T cells before it can eliminate all the tumor cells, then the tumor wins. And so the solution to that seemed to be simple. You just put in an antibody and disable the brakes for a while, and you can keep the T cells rolling for as long as you want to. And so you notice that the tumor cell isn't in this picture. And so this strategy for cancer therapy has nothing to do with the tumor cell. And this was bad, and then it was hard to convince people that you could treat cancer by ignoring the cancer cell, which caused us problems. But it's good because you can think, well, maybe this approach should be able to cure any kind of cancer, because the T cell itself doesn't know a kidney cancer from a bladder cancer. It doesn't know a KRAS tumor from any other kind of tumor. It just knows there's a cell there that shouldn't be there and has a bunch of stuff in it that's not right. And so that's one really compelling reason to do this. Again, it could be a universal cancer treatment. And the other one is, perhaps not so obvious, but since this is started by tumor cell death, this cross-priming, then basically anything that kills cancer cells will prime an immune response. It won't be effective unless you block CTLA-4, but then what you can do is start thinking about combining it with chemotherapy, combining it with radiation, combining it with targeted molecules, again in a way that you don't necessarily have to think about killing every last tumor cell with whatever your more conventional therapy is, just enough to get the tumor's immune system activated and take them out and then give you memory. And so, fine idea, but does it work? This is one of the first experiments that we did using a transplantable tumor. Again, it grows on mice. You block CD28 and it grows faster. So there is an incipient immune response going on, but it's not sufficient. But if you give anti-CTLA-4, it grows for a while and then goes away. And when the postdoc, Dana, came in and showed me these results, I was flabbergasted. I said, this is, as you heard, I said, this you're covering up this one molecule and tumors get rejected and they're immune. I said, that can't be. You got to repeat the experiment. So he told me he was going to Europe to see his girlfriend over the Christmas holidays. He'd started when he got back. I said, no. You start the experiment now. Don't tell me what's what until you come back, and I'll measure the tumors on the mice. Anyway, it did reproduce, and we did it in a lot of other kinds of mice and a lot of strains of mice just to make sure it was generalizable. And basically what we found is tumors like this one, which is a carcinogen-induced tumor, these that have a lot of mutations are very strongly immunogenic anyway, were readily treated by just monotherapy. Others, which are more poorly immunogenic, CTLA-4 by itself didn't do anything. As you can see here, this is the B16 melanoma. It was thought to be completely non-immunogenic. Glenn Dranoff, on the other hand, had shown that if you transduce those B16 cells with the genes for the cytokine GM-CSF and then irradiated them, you could use that as a vaccine to get prophylactic immunization. But that also doesn't work. That also doesn't work if the tumor's on board here. But we saw Glenn's results, and GM-CSF, what it does is activates and enhances that cross-priming process. I said, well, this is the perfect match. You have something that enhances cross-priming, and you have something that sustains the fruits of cross-priming. So maybe if you combine them, they'll work better together, and it's a true synergy, because 0 plus 0 gets you close to 100%. In some experiments it was, but it was never less than about 85 or 90% or so. And we've done this hundreds of times. And so what's going on in the tumor? Well, this is the untreated tumor. You can see the vasculature here in red, T cells in green, the odd T cell out here. If you look at the kind of T cells, many of them have purple nuclei here. That's a master transcription factor called FOXP3 that turns these guys into suppressor cells, regulatory cells, that make TGF-beta and IL-10 and other things that keep T cells from coming in and keeping them from functioning, even if they get in there. But if you give CTLA-4, in this case together with a vaccine, you activate the vasculature. You see it expresses this integrin ICAM, another one called VCAM. That allows the T cells to extravasate where it's expressed, and they go through, as you can see here, see them doing it, they go out into the tumor where they also then proliferate. If you're the kind of T cells, you've still got some of these suppressors, but they're vastly outnumbered by FOXP3 negative CD4 cells making TNF alpha and gamma interferon, both of which can kill tumor cells, as well as CD8 cells, which can degranulate and put things into CD8 cells that directly kill them. And so about 2 weeks after this picture is made, that mouse will have to be euthanized because the tumor will be too big. 2 weeks later, this mouse will just have a white spot where the tumor used to be, because one of the targets of T cell is tyrosinase-related protein 1, which is involved in melanin synthesis. So the cells in that area are killed by these potentially autoreactive T cells. So I won't go through this. Oh, by the way, those kinds of tumors that we have to help are the ones that can be spontaneous tumors that have very low mutational loads. So we teamed up with Metarex, who have these mice that have their immunoglobulin genes replaced with human. So it made a fully human antibody CTLA-4. That's actually an underestimate. It's probably over 100,000 now. Their objective responses in early trials, including melanoma, prostate, kidney, bladder, and other kinds of cancer, as we would have expected. There are adverse events. Most of them, about 40% of patients don't have any adverse events whatsoever. 60% do. The vast majority of them are things like hypophysitis, colitis, pneumonitis. Anyway, these itises that generally can be treated with systemic steroids, and then you wean the patients off and they go away. Now that it's in widespread use, there are incidents in a very small fraction of patients of type 1 diabetes and things like that. But it's generally manageable and certainly fewer side effects than with most chemotherapies. And so this is one of my favorite slides. This is a woman named Sharon, who is a patient of a good friend of mine, Tony Rebus. And in 2001, she had melanoma from playing sun and being out in the sun playing tennis in Southern California. Anyway, you can see this lung mass here, pleural effusion here. She'd failed everything, a couple of chemotherapies, dendritic cell vaccine, and high-dose IL-2. Tony asked her if she wanted to be on phase 1 of this new drug, ipilimumab. And she said, sure, I just want to live a few months. Just you just keep me around to see my kids graduate from high school. So that was in 2001, about 6 months after that single dose. Her tumors went away. I visited in 2011, 10 years later, and Tony introduced me to her. She'd come in for her first decade checkup, and she's actually still doing fine at 16 years after a single treatment. And so there were 2 other patients in that phase 1 that responded. Anyway, after a lot of smaller trials, there was an 800-patient placebo-controlled randomized trial of ipilimumab in metastatic, that is stage 4 and 3 melanoma. And this is the control here. You can see the median survival of patients diagnosed with melanoma at the time was about 11 months. And again, no drug had ever improved survival in a randomized trial. But you can see ipilimumab increased it by about 3 months, the median survival by about 3 months. And so it would have been approved by the FDA just based on that. But the fact is, between 2 and 3 years, the survival curve flattens out and stayed flat for the 52 months of this trial. The endpoint was overall survival. So that means after about 3 years, nobody's dying of melanoma anymore, and virtually nobody. And so that was repeated, or rather, there was a 10-year follow-up of almost 5,000 patients that had been, for whom there were records available in 2014. And what you can see is, again, in those patients, it also flattens out about 3 years. And 22% of patients are alive 10 years after a single round of treatment, which now is 4 doses of 3 mg per kg at 3-week intervals, and then it's stopped. And so, of course, that's pretty significant. Those people basically, by the way, are essentially cured. Virtually no one relapses after they've had these responses. There are some, but the number is very, very low. And I know quite a few, and they certainly consider themselves cured and don't worry about it coming back anymore, which is a pretty healthy thing, given what it used to be like. Anyway, but why 22%? What about all the other patients? Can't we do better? Well, first of all, why is it just 22%? Well, there's many reasons, one of which is that maybe the antibody isn't on board when this priming event occurs. It's theoretically possible. But of course, the more interesting possibilities, there may be, are there other checkpoints that might be in play here? And so about the time these phase 1 results came out, the group at Dana-Farber, Tosco Hanju had cloned the PD-1 gene many years earlier. He didn't really know what it did, but then Gordon Freeman and Arlene Sharp cloned 2 ligands for PD-1, ligand 1 and ligand 2. So it's another checkpoint, but it's different than CTLA-4 in 2 ways. One of those ligands can be expressed by tumor cells by gamma interferon. So the tumor-specific T cell comes over, makes gamma interferon, trying to kill the tumor cell. The tumor cell puts up PD-L1, which through PD-1 then exhausts the cell, keeps them from functioning. And the other difference is that PD-1 targets the antigen receptor, and so there'll be more about this later. Anyway, just PD-1, there was a phase 1 trial in 2012. It moved relatively quickly after the success of anti-CTLA-4. A small trial with many different kinds of cancer. Very good responses in melanoma, about 28% objective responses. Also in non-small cell lung cancer. Also in renal cancer. There were non-responders, though. There were no responders in colorectal cancer. Although we know now that there is a subtype that have genomic instability, have microsatellite instability. They have a lot more mutations. Bert Vogelstein showed that they have a lot more mutations. These patients respond quite well. But also there were no responses in colorectal castrate-resistant prostate cancer in a small trial. And at the immunotherapy platform, Pam Sharma has figured out, I think, why that is. And I'll talk about that later. So where do we go from here? Well, you have these 2 checkpoints, both of which give responses in a fraction of patients. So maybe you could put them together. And so in my lab, we did this in some animal models, and what you can see is you block CTLA-4 plus PD-1 or its ligand, you get an additive response. So it looks like they can complement each other. It was known very shortly after this that there were patients that didn't respond to CTLA-4 blockade, did respond to PD-1, and vice versa. And so BMS put together a big trial. This is a phase 1 of combining the 2 and giving them concurrently. And in this trial, again, this is metastatic melanoma. 2/3 of the patients had tumor shrinkage. Half of them had objective responses. You can see here these spider plots of individual patients. So about a 50% objective response rate. But this was approved by the FDA in 2016. There's 2-year follow-up that was talked about a year ago, and 60% of the patients are alive at 2 years. And so we obviously don't know yet, but if the ipilimumab effect on survival dominates. You know, if those guys make it 3 years, the chances are they'll be good to go, you know, for a long time. And so this really shows that you can achieve these amazing things, not just in melanoma, as I showed you, but in other kinds of cancer as well. But it'd be nice to move this forward. In order to do that, we really need to know a lot more than we do now about the cellular molecular mechanisms that are involved. The clinical stuff is going so fast, the science can't keep up. And we really need, if they're going to be rational combinations, need to know how they work so that we can, in a rational way, put things together, be there other immunological treatments or standard of care or the small molecule inhibitors. We need to know both directions what the impact is on the immune system. Design to target new molecules to improve efficacy. We have identified another one that could be targeted in an agonistic way that's a positive co-stimulator called ICOS that I'll talk about in a little while. It'd also be great if there would be predictive prognostic or pharmacodynamic markers. Despite a lot of work, it's very difficult with baseline samples to predict who's going to respond, but there is considerable progress being made on signatures in on-treatment patients very early on that can allow you not only to predict who's going to respond, but the next step to take in therapy. I don't have time to talk about that today, though. So this slide, this sort of slide, gets a lot of play these days because one of the things that's become apparent, first of all, just from cancer biology, is that different kinds of tumor differ according to their mutational load. Those at the top are melanoma, of course, from carcinogens, lung from carcinogens, stomach and bladder cancer from carcinogens, and then, sorry, melanoma from sunburn, the carcinogen effect of ultraviolet radiation, and these others from chemical carcinogens probably. And so this was a study, the paper that Tony Schumacher and Bob Schreiber worked with. They adapted this from a cancer biology slide. But it's also apparent that kidney cancer, for example, responds very well, even though it's way low before where you expect it was. So this mutation load is not the only thing. There's other things that come to play that we need to know. But when you get down here towards pancreatic cancer, which you heard a lot about from Tony, and prostate cancer, you know, they don't, there's an occasional response, but that doesn't work very well. So these are two cancers that Pam Sharma and I in the immunotherapy platform are trying to really tackle now to try to see what we can do the further to the left you get on this slide. And so I'll talk a little bit about what we've been doing on prostate. So the first thing I should point out is that acetyl-A4, as I mentioned, there were some responders that had complete responses. Metastases go away, PSA going to baseline, but not many. And this was a phase 3 trial that was published in 2014 that just missed significance, so not very many responses. And as I told you, PD-1 and there were no responses in a phase 1 of patients who received this in a Bristol-Myers Squibb trial. So we started, Pam's Sharma's the PI on a trial using a combination of acetyl-A4 and hormones. But these patients are actually getting we do a biopsy and get tumor samples before they started ipilimumab. And so then we did genomic sequencing with Andrew Futrell's help and RNA to identify mutations. And the idea is to see what happens and then try to identify the antigens. So we're in the process of identifying the antigens, but what's come out already is an interesting sort of picture here. Is that there are two typical prostate cancer, we call them, that didn't respond. These had very low, in fact, no expressed non-synchronous, non-synonymous mutations. We've had 4 patients who had the typical, let me go here first. There are also 2 patients that had high levels of mutation relatively that were non-responders. This is small cell, it's a kind of cancer that's very hard to treat with any modality. So we don't know exactly what's going on there, but in these responders, we have 4 that have very low levels of mutations, expressed mutations compared to the other kinds of cancer, and yet they responded. So we don't know whether that's because the antigens, there are enough, there's one antigen in that 8 or whatever, or because the actual response may be directed, of course, to prostate-specific antigen or prostate-specific membrane antigen, or these shared targets. But that's an important thing that we're in the process of figuring out here. But something else that we know is that tumors can differ. There's a lot of tumors types, such as melanoma and lung cancer, that are highly infiltrated with immune cells. We call those immunogenic or hot tumors. A lot of T cells in them, very often with PD-L1 expression, for example. So all you have to do is block the checkpoints and you could get a response. On the other hand, tumors, particularly those that have very low mutational loads, although not exclusively, are really poorly immunogenic and very little T cell infiltration. And so in order to really get responses out of them, you've got to get the T cells into them. And so, and before you do the checkpoints. So one of the things that we realized in this study is that you could do that with CTLA-4 alone. This was a patient had a biopsy before they were treated with anti-CTLA-4. And what you can see in the pretreatment, there's some T cells around the tumor, but not in it. But after treatment with anti-CTLA-4, the T cells go into the tumor. It's robustly infiltrated now. But if you look at the kind of cells, I think you can see this, there are a lot of CD8 cells in there now, you can see. But we know that patients don't usually respond. Well, why not? And that's because the T cells themselves now in this sample are expressing PD-L1. In fact, if you look, sorry, this just illustrates this. The PD-L1 on these activated T cells, very little on CD4 cells, but a lot of PD-L1 on macrophages in these tumors. And so if you look in the, if you look at the different types of cells, again, very small amounts, if any, on CD4s, a lot on CD8s after treatment with anti-CTLA-4, a lot on macrophages after treatment, and a lot of the tumor cells themselves. And so anti-CTLA-4 treatment, while it drives the T cells in, also induces the expression of these inhibitory molecules, and that's probably why it responds so poorly. The reason that PD-1 antibodies didn't work was because there's no T cells in there for PD-1 to work on, presumably, before you give CTLA-4. So we've gone to Bristol Myers Squibb. They'd shut down their entire prostate cancer program, but upon seeing this data, they opened it up again, and Dr. Sharma is going to lead a multi-institutional trial of the combination in prostate cancer. But that's not the whole story, because there's another inhibitory molecule called Vista that was discovered by Randy Noel. It's kind of a mysterious molecule, but it's known for sure that it inhibits T cells. And what you can see here is, again, after treatment with anti-CTLA-4, it's induced a little bit on CD8 cells, but a lot on macrophages again. So again, treatment with one thing that blocks one inhibitory molecule leads to expression of the others to compensate. In any event, we work with a company called Janssen that has an antibody to Vista. So if the CTLA-4 and anti-PD-1 don't work, we can be able to go in with the triple combination and hopefully get some action there. And so this just shows the induction. Again, it's mostly on macrophages, not on the tumors. Vista is but on a little bit on T cells and a lot on the macrophages, as I said. So I could go on and on about this sort of thing, but one of the things that has been happening in the last few years is that because of some differences between CD28 and CTLA-4, one is that PD-1, that CTLA-4 can take weeks to a couple of months to yield responses, whereas PD-1 is usually very rapid. And also, the adverse events associated with PD-1 seem to be much less than those associated, not less severe necessarily, but less frequent than those that happen with CTLA-4 blockade. And so there's been a tendency for everybody to say, well, we got 2 checkpoints, let's use the one that's, that causes the fewest side effects and works faster. But that's really kind of a mistake, I think, because these are 2 completely distinct drugs, as I'll show you in the closing part of this talk. The one is that CTLA-4 is hardwired. Again, its biological role is to protect you from your immune system. You know, it's the fail-safe switch that stops the T cells from just growing in. What I failed to mention is that Attack Max Lab and my own knocked out CTLA-4, and what happens in those mice is they're dead by the time they're 3 weeks old because they're just full of T cells, basically, in all the tissues. And so it's an absolute critical downregulator. Whereas PD-1 is induced resistance in the tumors, particularly. Again, CTLA-4 targets the CD28 pathway. PD-1 targets the antigen receptor pathway. CTLA-4 works during priming, we think. PD-1 works on exhausted T cells. CTLA-4, again, probably because of how it works, can bring new T cells into a tumor, where PD-1 doesn't seem to do that. It just expands the T cells that are in there. You get a lot more numbers, but relatively fewer new clones. So I'll show you in a few minutes, CTLA-4 predominantly affects CD4 cells, but PD-1 predominantly affects CD8 cells. As I showed you earlier, CTLA-4 Blockade can move cells into tumors. We have some kidney data that's a lot clearer, but PD-1 doesn't seem to be able to do that. And again, CTLA-4 leads to slow responses. PD-1 is usually is much more rapid and is RECIST compatible. For those of you who know what that means. Anyway, a thing that's also becoming apparent with time is that recurrence after treatment with CTLA-4 is rare. But with PD-1, it's significant. In melanoma, about 25% of patients relapse in 2 years or so, and in lung cancer, about 35%. And this is very often due to mutations in the gamma interferon pathway or loss of beta-2 microglobulin, which means no more expression of CD8 or whatever. And for reasons that aren't entirely clear, that really doesn't happen after response so much in CTLA-4 treated patients. And so one of the things that we're trying to do is really figure out in detail the cellular mechanisms of these two drugs. And the way we're doing that, and this is really work in progress now, is the use of CyTOF, where you can do 43 parameter analysis. Most of you probably know what this is, but it's just flow cytometry, except that you use heavy metal ions instead of fluorescent probes. So what we do is take tumor cells, different tumor cells, stain them, again, with 43 parameters. They're not all colors. We have some channels that we use to barcode the samples so we analyze them together. Some channels used for BRDU so we can inherently know which cells are proliferating and some other things. But this is this is the MC38 tumor, looking at all the cells in there. These are CD45 positive cells, but you can see all the different kinds of hematopoietic cells in there. Dendritic cells, myeloid cells, anyway, all the different kinds of cells, and do this in an unsupervised, unbiased way. And then the hope is to then use meta-analysis of the associative changes in those different populations with changes in tumor size to identify those populations that are actually identified with tumor rejection itself. And so this is the results of this sort of analysis of the MC38 tumor. This is a colorectal carcinoma. It's very highly immunogenic. And so it's sort of protocol is this. We give the tumors to mice, treat them with anti-CTLA-4, and on day 13, before they've started being rejected, but just about the point where they're going to harvest the tumors and carry out the high parameter analysis. And so this is actually, if you look just at infiltrating T cells, so these are CD45 plus, CD3 epsilon plus, so just gating on those. And so this is what mice treated with a control, with an isotype control, look like in contour plots. And so if you compare this just by eyeball with CTLA-4, I think you can see some interesting things. The first is that this population goes down a lot when you treat with anti-CTLA-4. This population goes up a lot relative to control. This one really doesn't change too much. And these here some of those go up, but not all of them. With PD-1 treatment, anti-PD-1 treatment, relatively little happens to this population, or this population, or this population, but there are increases in several of the peaks here that really, some of them overlap, but some of them don't overlap with anti-CTLA-4. And you can see that better here. You see almost loss of red, the CTLA-4, in this population, a big increase here, and then kind of mixed here, and no real change here. Well, what are those different populations? What's happening here? Oop, wrong button. Nope. I'm losing the ability to advance these. Ah, there we go. Anyway, so now we let you know what these are. These are regulatory T cells. These are effector CD4 T cells. These are CD8 T cells. And these are NKT and gamma delta T cells down here. So again, these increase with CTLA-4 treatment. Those decrease with CTLA-4 treatment. And the two antibodies enhance different populations of those. So again, I don't know what's going on with this. Ah, okay. Just takes 6 or 8 pushes, I guess. Anyway, so what you could do is cluster these. By the way, those groups are arranged by just the computer puts with Visney software cells that are more like each other together in clusters. And so you can identify the discrete clusters, which are shown here, and then these are heat maps of the different markers that they express there. And so you get a lot of resolution. In this case, it's a very T-cell biased panel that has transcription factors and surface markers for all the known types of T cells. Okay. So this just sort of breaks them out into different kinds to make the points easier. So these are FOXP3 positive regulatory T cells here. And what you can see is there are 2 different types, a KLRG1 positive and KLRG1 negative. This is a minor population. There's relatively more of this population there. See the blue dots. But it's obvious that CTLA-4 treatment depletes a lot of those regulatory T cells. This is something we've been seen before in mice, but I can tell you we don't see this in humans. So there seemed to be a fundamental difference. But anyway, that tells us something we already knew. If you look at what happens with CD4, thank you. Right. So we need the pointer here. So anyway, if you give anti-CTLA-4, what you see is there's a huge increase in this population. Now these are very interesting cells. These look like follicular T helper cells. They express a molecule called ICOS that's really only been found before, before they were found earlier. But this is a peculiar population because phenotypically it looks a lot like follicular T helper cells, which are in the lymph nodes and spleen, and they make type 2 cytokines that help B cells class switch immunoglobulins. There's also a kind of Treg that has ICOS. Neither one of those cells would do you any good in a tumor setting. In fact, they'd probably hurt. But Pam Sharma, about 10 years ago, in studying what happened in bladder cancer patients after anti-CTLA-4, found that phenotype of cells and showed that they make gamma interferon and TNF-alpha in response to tumor antigens, and their number correlates in the blood, even correlates with survival after CTLA-4 administration. And then we collaborated and showed that if you give an agonist signal through ICOS, it makes the T cells much more active. It makes them proliferate a little bit more, but it induces more T-bet into them. It makes them a much more Th1-like T cell. And so that's basically what those cells are. As you can see, they don't go up with PD-1. Then there's these other cells that don't go up with any of them. But there are also some CD8s that go up with anti-CTLA-4. These, but not so much PD-1, those and those. And these that go up with both, really, a little bit more perhaps, CTLA-4. But these are the primary ones that go up with PD-1. And these are PD-1 high and TIM-3. So these are the most exhausted CD8 cells. And they've got T-MET also, obviously. But these cells are the ones that are probably making, killing tumor cells, making gamma interferon, but they got stopped by the PD-L1. So if you block that pathway, if you intercept it with antibodies to PD-1, those cells could proliferate, and we know they're proliferating from one of the channels of cytol. So that's basically what you'd expect from the model. But what this suggested, because they still have PD-1 on them, is that if you quit giving the antibody, as soon as the antibody goes, those cells are going to become exhausted again and are not going to be able to sustain the anti-tumor effect, which may, we don't have any data for this, but may explain a little bit the lack of durability of that. There's a paper from actually two papers from two different groups recently showing that once CD8 cells reach that particular cell surface phenotype, there are epigenetic changes that prevent them from losing those. So they're basically locked in. Anyway, this data is consistent with that, and you get these other populations as well. So we also did this experiment with the B16 model together with the GVAX. So this is a very poorly immunogenic tumor with a vaccine, and so the reason we did this is to see if we could pick out the same or would they be different mechanisms. And so there was a difference in size, and so that's important because later on we could look at the individual mice and correlate the changes with the role of those mice, with the role of those classes. So anyway, this is what the map looks like of the B16 cells treated. Well, this is just the overall map again, sorry. The T cells in there. And then just going directly to the types of the cells. These are these Th1-like cells here. These are those ICOS, PD-1 high, TIM-3 high exhausted cells here, and some of the other ones there. So anyway, the main point, not spend too much time on this, is the main signatures are exactly the same in this poorly immunogenic tumor with a vaccine. And so then, as I said, we could go back and pick out those different clusters where we know exactly what's expressed. And then because we kept track of each sample, each mouse that was analyzed, we can do a meta-analysis. And so what it shows, for example, is not surprising, these Tregs correlate positively with tumor size. So these are inhibitory populations, so the more of these cells, the bigger the tumor gets, which is what you'd expect. If you look at these Th1-like cells, you can see they negatively correlate. Sure, these are really playing an obvious role, correlating with decrease in tumor size. But the other CD4 population that was ICOS negative really doesn't correlate one way or another with anything. So the action is with these cells here. And if you look at the CD8 population, these are more complex, obviously. These, of course, are the most exhausted cells. You can see here they correlate negatively. This is another population that's similar, maybe a little bit more exhausted, partially exhausted, but they express EOMES, which is a terminal differentiation antigen. These don't. And it's a marker that tells you that those cells are terminally differentiated. They both correlate with tumor shrinkage. These cells don't correlate one way or another. And these cells, which appear like they're unactivated, these may just be T cells that are passing through the tumor. We don't know, but they're not active. But they actually have a slight negative correlation, so they're actually getting in the way of the other cells. Maybe it's my thumb. Or maybe it's okay. Any event, so what this tells us, I think, is that the therapeutic mechanisms of CTLA-4 and PD-1, anti-CTLA-4 and anti-PD-1, are quite distinct. In fact, they're almost completely non-overlapping. They may explain why, when you put them together, it's such a powerful combination. Different subtypes contribute. And I'm not showing you, we also try to look at correlation of just CD4s and just CD8s. And correlating with these subgroups is much more predictive of what you're going to see than just looking at all CD4s or all CD8s, for sure. And I can tell you we've begun to study human infiltrates. We're not just far along, but it looks like the same. It's exactly the same populations that are involved. After all, those ICOS positive Th1s, again, I'm sure I'm identified in patients several years ago. And so I think this gives us some clues as to ways to fine-tune what we do, and more ability to really know the mechanisms and watch for things in order to build rational combinations. And so I'll just sort of wrap it up here. That's not the end of it. We've talked about some of these molecules, but TIM3, which I mentioned, it's another immune checkpoint. LAG3 is another we didn't talk about. There's another one called TIGIT. There are at least 8 or so more of these guys that inhibit T cells. We don't know, if they'll be important or how important they'll be. But a lot of work's underway both in mouse models and in the clinic in trying to evaluate whether these could be used as combinations. But in moving forward, I think, we don't understand this. Anyway, is combinations to do it. So I've shown you an example of blocking multiple checkpoints. We've also done some things with positive checkpoints that I have a chance to talk about. Combining, as I mentioned earlier, combining conventional therapies with checkpoint blockade makes a lot of sense. There's beginning to be a little work there. Everybody thinks instinctively, well, chemotherapy is immunosuppressive. Well, surprisingly, it isn't always. And even if it is, typically those drugs have half-lives that can be measured at hours. So you can just give the chemo again enough to kill some cells, stop, and then start the checkpoint as soon as you can. There are other immunosuppressive factors, IDO, for example, that can be blocked as well. Local ablation works. We've shown that cryoablation of one tumor followed by CTLA-4 could give you systemic immunity. It'll take out all the tumors in a mouse, as will an oncolytic virus in one tumor. You can eliminate one tumor and get systemic immunity if you follow it with that of CTLA-4. That has the advantage that the RNA oncolytic virus that we've been using enhances the innate immunity by getting type 1 interferons produced, which enhance cross presentation. So that can be important. And then vaccines, both the shared type and individual, might become more powerful. The individual here, there's quite a bit of activity in identifying the neoantigen in a tumor-specific way, those neoantigens, and it's in the clinic with some pretty promising results already in very small trials. And finally, the genomically targeted therapies, I think, can also be, and that's beginning to be done, combined with these immunotherapies. What you have to know, though, is really what their impact on the immune system is so that you can combine them rationally. And what people are beginning to do, at least in chemotherapy and in radiation, are beginning to, again, think about ways of changing how they view the desired impact of their therapy. Do you want to kill every last tumor cell? Or do you want, with the radiation, for example, and run the risk of after your first treatment, the immune cells come in and you kill them with your second treatment, which would be, not take advantage of it, or just, spare the destruction of the immune system by giving closely spaced multiple doses, which is what my group in New York is really doing at this time, to just make it friendly with immunotherapy. And I think that will have to happen with the genomically targeted therapies. Instead of giving them chronically, perhaps dose with a high level of them and then start the immunotherapy, and you've still got a chance then maybe of the targeted therapy working, at least not giving you resistance. If the immunotherapy doesn't work, then you could start over and not get resistance yet. So a lot of things to be done yet, but it's a promising time. And so I think the way that this has changed cancer treatment is significant. I mean, I think when we started this work, I must say immunotherapy was held in disrepute throughout the tumor biology world for sure. But now I think it's established as a legitimate and very powerful way of obtaining very durable responses, albeit in a fraction of patients right now. But this is the way that we used to look at cancer therapy, is you treat a large number of patients with the drug, then look at changes in median survival, and hopefully, you get some people that survive longer. What we know now from ipilimumab, for sure, is that in melanoma, you can get a fraction of patients, 20%, that will be alive a decade later. And this next one is not, if it works, this is an aspirational goal, obviously, not anything based on data, but we know we can get up to about 50% or so in melanoma, again, by just combining anti-CTLA-4 or anti-PD-1. And obviously what the goal should be is to do whatever combinations you need to raise the survival tail as high as you can get it, and do this in as many different kinds of tumors as you can get it. And that's quite a challenge. There's a lot to do, but the good news is that I think we know the fundamental rules. We're beginning to learn them. We know a lot of the pieces now, and so we just need to be very careful now, do a lot of basic science, know what we're doing, and then make the combinations. And I think that we'll be able to do this, at least in some kinds of cancer, in the very near future. And so I'll stop there, and thank you for your patience.
So we thank him, Jan, for so elegantly showing the power of basic knowledge to actually transform cancer treatment, which was also earlier shown by Tony's lecture. So I think that we are very happy as the first prize winners for the Sjöberg Prize to be able to honor Tony Hunter and Jim Allison.